So the work that I presented was several different mass spectrometry studies that we did on hematopoietic stem cells, either from normal mice or from mice harboring a loss of function mutation in the TET2 gene. And the reason for us wanting to look at the proteome is that historically this has been very much overlooked in hematopoietic stem cells because mass spectrometry-based approaches require very large cell numbers, and hematopoietic stem cells are very rare within mammalian tissues, so it has kind of taken a while for the technology to catch up. But recently there's been advances both in low-input mass spectrometry-based proteomics and in ex vivo expansion of hematopoietic stem cells, which has made this much more feasible. So we have combined both of those approaches and characterized the proteome of freshly isolated wild-type and TET2 knockout hematopoietic stem cells, as well as the secretome and proteome of ex vivo expanded hematopoietic stem cells, and looked to some extent also how it correlates, or rather anti-correlates, with what we see in transcriptomic data. So for example, in the TET2 knockout hematopoietic stem cells, the freshly isolated, we found that extracellular matrix proteins were differentially expressed between the wild type and the knockout cells. And when we looked at the expression of those same, when we looked at the transcripts of those same proteins, we found that they were either unchanged on the transcript level or showed completely opposing expression. We then went on to show that if we cultured the hematopoietic stem cells in hydrogels functionalized with these extracellular molecules, that the interaction with these differentially affect the hematopoietic stem cells depending on if it's a wild type or if it's a TET2 knockout cells, proving that there is a lot of functional relevance in data that can only be picked up by proteomics and not transcriptomics.